Hello everyone. Welcome to today's live broadcast, exploring the stoichiometry of macromolecular complexes using multi-signal sedimentation velocity analytical ultracentrifugation by Dr. Chad Brautigam. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman XL-A, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman XL-A. Beckman XL-A is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman XL-A products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmanxla.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the slide. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button. I would now like to introduce today's speaker, Dr. Chad Brautigam. Dr. Brautigam earned dual undergraduate degrees in chemistry and biochemistry in molecular, cellular, and developmental biology at the University of Colorado, Boulder. He then joined Thomas Steitz's lab at Yale University, where he studied the structural enzymology of DNA polymerases. Following the award of his PhD in molecular biophysics and biochemistry, Dr. Brautigam's postdoctoral work was in the laboratory of Johann Diesenhofer at the University of Texas Southwestern Medical Center. While in Dr. Diesenhofer's lab, he determined the three-dimensional structures of proteins involved in neuroplasticity and blue light sensing in plants. It was at this time that Dr. Brautigam first developed an interest in analytical ultracentrifugation. In 2007, he joined the faculty of UTSW, advancing to the rank of associate professor in the Department of Biophysics in 2012. Dr. Brautigam is also the director of Macromolecular Biophysics Resource, a core laboratory offering training in and access to a variety of biophysical techniques. He has served as an instructor at AUC workshops around the world for over 8 years. Additionally, Dr. Brautigam developed software for biophysical data processing, analysis, and presentation. These include Redate and Goosey, which both act on AUC data. Before we get started, I would like to ask a few poll questions. Our first poll question is, what instrumentation do you currently use to determine particle interactions, particle size, and heterogeneity? You can answer multiple questions here. Analytical ultracentrifugation, the topic of the day, surface plasmon resonance, dynamic light scattering, nuclear magnetic resonance, or others. We'll take a few moments and we'll let the answers populate. Okay, just a few more seconds. It looks like about 35% use analytical ultracentrifugation. 35% also use dynamic light scattering. NMR, or nuclear magnetic resonance, comes in at 8%. SPR, or surface plasmon resonance, comes in at 4%. And there are other techniques as well that are coming in at 19%. One additional poll question before I turn it over to Dr. Brautigam. For what types of analyses are you interested in using analytical ultracentrifugation? Again, multiple answers are allowed. Heterogeneity, reversible and non-reversible heteroassociations, self-associations, molecular weight determination, and shape and size determination. We'll take about 10 to 15 seconds to allow everybody in the audience to answer. Great. Looks like 31% are interested in shape and size determination. 28% are interested in heterogeneity. 22% in just molecular weight determination. And at 9.4%, we have two results at reversible and non-reversible as heteroassociations and also self-associations. All of these methods are capable in the analytical ultracentrifuge. Thank you, everybody, for your answers, and I would now like to turn it over to Dr. Brautigam for his presentation.
Hello, everybody. Thanks for tuning in, and thanks to Chad for the introduction. As he mentioned, I'll be speaking to you today about the technique of multi-signal sedimentation velocity. I won't go over my title again. So I just want very briefly, first of all, to thank the organizers for letting me speak to you today. Those would include the people at Beckman XL-A, particularly Lindsay Schwarzkopf and Chad Schwartz, and the people at LabRoots who've helped out with the technical aspects of getting this to you, mainly Don Cruikshank. So I just wanted to go over with you what the goal of a multi-signal sedimentation velocity experiment is. First of all, generally we can say that the goal is to resolve amounts of materials that are hydrodynamically not resolved. Specifically then, in an analytical ultracentrifugation experiment, what we're really talking about there is that things that have the same sedimentation coefficient. Now, for most of you who have tuned in, what this really probably means is that you want to figure out the relative amounts of species in a co-sedimenting complex. In other words, you have two species that are interacting and moving together as one entity during the sedimentation velocity experiment. So together with multi-signal sedimentation velocity, we get both molar ratio and hydrodynamic information on size, shape, and molar mass, and that allows us to get what I would call the true stoichiometry of the complex. What I'm saying there is, let's say, distinct from stoichiometry you might get as the, let's say, n value from a calorimetric experiment. For instance, let's say you get 2 to 1 from that experiment. That might be actually 4 to 2 or 6 to 3, and that might have profound implications for the mechanism of your complex. And so you want to know that, and multi-signal sedimentation velocity is an excellent way to get at that, particularly if you're interested in modeling your complex. And so on this slide I show basically a standard sedimentation velocity experiment. Over here on the left we have the data. And you'll see that the data are presented as data points. Purple here is showing early time scans, red showing late time scans. And there are actually fits to these data shown as solid lines. If we do a standard CFS analysis, then we get the CFS distribution shown over here on the right. Now, that shows us that there's one thing there sedimenting at about 5 Svedbergs. What isn't told in this distribution is that there are actually 2 things present. But from a single signal, there's no way for us to decompose this CFS distribution into into its component pieces. Okay, so on this slide then I show the same data actually taken at 2 different signals, and you can see that there's a amplitude difference in the signal. If we do a multi-signal sedimentation velocity experiment, then what we see is the distribution on the right. So now we get 2 two distributions, one for, let's say, protein A, and the other for, let's say, protein B, one red, one blue. By knowing the signal increments of protein A and protein B, and by collecting data at two signals, we can decompose that C of S distribution into component distributions, and knowing the molar ratio from these C of S distributions and the mass information that we get from the analytical ultracentrifugation experiment automatically, then we get this magical quantity stoichiometry, which is what we're after. So in this webinar, I'll be going over some basics of sedimentation velocity. I think most of you are probably pretty familiar with this stuff, but we're going to be talking a lot about how the multi-signal sedimentation velocity analysis works, and we need to all be on the same page. Then I'll talk about specifically the theory of multi-signal, how to run an experiment, how to analyze the data, and then throughout there will be examples, and at the end we'll talk about a problem case that we worked out. All right, so this animation then shows us the phenomenology of sedimentation velocity. You can see at early times we have a flat purple line, and then at late times we've got a flat red line, and we have this data trace going from left to right across your computer screen there. And so what we're seeing is a certain velocity of the molecules moving toward the bottom of the sectorial centrifugation cell. This region between where there is solute and where there isn't solute is called the boundary. And so we're watching the boundary move toward the bottom of the cell. So from its velocity, we get the sedimentation coefficient. But you'll also notice that these boundaries start flattening out as we get later and later time. That's actually a measure of the diffusion coefficient. And so from such an experiment, we can get both the sedimentation coefficient and the diffusion coefficient. This is the Svedberg equation I'm showing here. So the rest of the quantities here are experimental quantities that we either know or should know. The gas constant and temperature, the partial specific volume of the protein, and rho, which is the solution density. Since we know all those things, then we automatically get molar mass out of that. So from a sedimentation velocity experiment, we get sedimentation coefficient, diffusion coefficient, often encoded in terms of the frictional ratio, and molar mass. Okay, so these are the species properties that we might get from a sedimentation velocity experiment. The equation that governs basically what the data looks like is called the Lamb equation, and that's shown here. It's a partial differential equation. And you can see that we have our old friends diffusion coefficient and sedimentation coefficient shown in there. There's some other things in here: radius from the center of rotation, the weight concentration of the species, the angular velocity of the rotor. And so when we put all that together, we get this over here on the left, which is basically the change in concentration as a function of time. And so that's what we're going to be looking at when we look at solutions to the Lamb equation. Just in case you're curious, basically there are no exact analytical solutions to the Lamb equation. But it's pretty easy to solve it numerically, and in fact every program that I know of that does the task of solving the Lamb equation does it numerically, and it uses this Clavery finite element approach, which is briefly shown here. I don't want to belabor this too much, but you can see that what we get here, our result, is concentration as a function of radius and time. That's going to be the sum of a bunch of these triangular hat functions. So those are shown here. They are just triangular functions. If we add them all up, let's say in the beginning we would have a uniform distribution. If we added all those up, that would be a straight line right across the board. But at a later time, we'll be scaling them by these h factors here. And then at a later time, those h factors times those hat functions would look like this. When we add all that up, what we end up getting is something that looks like that, which is basically something that looks like our data. And so all data are modeled with these numerical solutions to the Lamb equation. So this is a solution to the Lamb equation starting from unitary concentration. Basically, this was a simulation for a species that had a sedimentation coefficient of 2s and a diffusion coefficient of 2.3 fix. You can see again shown in purple we have early time points, and in red we have late time points. And each one of these stands then, or is basically a moment frozen in time. My point here is that the Lamb equation holds for all radii, so we've solved it for all radii, and it holds for all time. So we've solved it for all times and then just displayed discrete times shown in this graph. Now the data basically are going to look like the figure on the left. What we can do is basically fit Lamb equations, solutions which look like this, to our data. And that's actually a fairly straightforward thing to do. Basically what we end up doing is solving the Lamb equation with sedimentation coefficient and diffusion coefficient as fitted parameters, and then we scale it by a concentration parameter, which basically allows us to fit it into signal space. So I point out here that data are in signal space, so our solutions to the Lamb equation have to be transformed into that space with this C factor here. ART is going to be our raw data. And so these data that I'm actually showing are not simulated. This was actually for an experiment I ran back in July. And you can see that the sedimentation coefficient refined to 3.2, the molar mass, which is related to the diffusion coefficient, of course, refined to 39,678 grams per mole. So we can actually use these Lamb equation solutions to fit the data directly in data space. Now, sometimes we have data that are a little bit more complicated. For example, here we have two boundaries, and just in case you're not used to looking at data, you have to look at the later time points. One of the boundaries is there, the other boundary is there. So we have a fast-moving component and a slow-moving component. For a complex mixture of species, then what we use is a bunch of Lamb equation solutions. So what we're going to do is, instead of using a single concentration factor to scale our Lamb equation solutions, we're going to use a distribution of them called the C of S distribution. Most of you are probably familiar with this. This is the go-to method for the analysis of sedimentation velocity data. So we have our data in a data space. We have a C of S distribution, which must be relating a bunch of Lamb equation solutions to that data space. And operationally in the program, what happens is we discretize this integral, and we have a resolution, shown as a little m here, of a bunch of C values, and those are all scaling a bunch of respective Lamb equation solutions. What this ends up looking like is the distribution shown over here on the left. So if we have a distribution that looks like this, we scale this times the respective Lamb equation solutions, add it all up, and it ends up looking like the solid lines here, which are the fits to the data. So the C of S distribution is basically a distribution of more or less concentration factors, those signal concentration factors, that are scaling Lamb equation solutions to fit our data in data space. Okay. Now, an interesting aspect of the C of S distribution is that it's normalized such that it is differential. So if we integrate this peak in our C of S distribution, what we end up getting is the signal concentration of that slower species, shown by this arrow over here. So it's basically telling us how much of this 5.5s species is present in the signal, okay? And it would give it out in signal units. In other words, it would give it out in absorbance units in this case. Okay. So other important aspects of the CFS distribution, again, basically, it is regularized, so, and that just allows us to have a physically reasonable distribution. In other words, it suppresses high-frequency oscillations in the distribution that are... Okay, so if you have an XLI, then you can always also use laser interferometry to track the signals. Therefore, you have access to either 3 or 4 signals. Okay. So how can we take advantage of this? Well, macromolecules will often respond to interrogation at different wavelengths characteristically due to their unique compositions. In other words, their primary sequences. In other words, for example, if we have proteins with different ratios of tyrosines and tryptophans, those would respond differently to different wavelengths, let's say 280 and 250, and basically those spectral signatures allow us to track components uniquely. As you might expect, we need as many signals as components. And so if we're tracking 2 things, which is going to be the majority of what I'm talking about today, we need at least 2 signals. The maximum number of components, of course, being tracked in a Beckman XL-A is 4, and I'm not actually aware of anyone having done 4. The maximum I've seen is 3. All right, so now in order to understand how multi-signal sedimentation velocity works, at least the approach that was pioneered by Peter Schuck, we need to re-examine the CFS distribution and actually make a new kind of CFS distribution. So we're going to be examining multiple signals. The multiple signals are going to be shown over here on the left side of this equation. Basically, we're going to be globally analyzing more than one signal, so we have A sub lambda there as a function of radius and time. And that's going to be a summation over all components present. So there might, let's say there are 2 components present, this large K here will be 2. We then have our distributions being multiplied by a respective extinction coefficient. So we need to get into signal space, and actually our distribution here is going to be in concentration units now. So to get the signal units, we need extinction information. These CKFS distributions will be scaling these Lamb equation solutions, which are similar to before, but they're respective to the specific component. All right, so some key points here. We must have at least as many signals as components K. The signals will be analyzed globally. The CKFS distributions overlap in S space usually, and we will have K of them. Okay, so again, in my examples, there's going to be 2 CKFS distributions, and they're going to be occupying the same parts of S space. Because we have access to extinction information, the CKFS distribution is normalized such that if we integrate the peak, it reveals the concentration of component K. This extinction matrix that I'm talking about must be non-singular, and I'll talk about that in a moment. Also in this treatment, hyper or hypochromicity is neglected. Okay. So just to illustrate these key components again, the key point 1 is that if we have 2 components, protein A and protein B, we need 2 signals, and these 2 signals are going to be analyzed globally. Here we see that our 2 CKFS distributions are occupying, basically overlapping in S space, in that if we integrate them, so if we integrate the blue one, we get the area underneath it, it's in this case 2 micromolar. The integral of the red distribution is going to give us 5 micromolar. So the molar ratio of these co-sedimenting species is 2.5 to 1. So now this actually gets to the what I want to call the difference between molar ratio and stoichiometry, and that is that this molar ratio is 2.5 to 1, but we're really not going to have 2.5 molecules bound to one molecule. So what this probably means is that this was a 5 to 2 or 10 to 4 complex, and you would get that information by knowing the monomolar masses of protein A and protein B, and then the molar mass that you got from the hydrodynamic information that we get from doing the analytical ultracentrifugation experiment in velocity mode. Okay, so now I want to talk about this matrix of extinction information. So we have this matrix. If we have two components, then the extinction of component A at the different signals is shown by this column vector here in that matrix, and the extinction information for component B is in that column vector here. So if we have a Cartesian space here, where we have the extinction at wavelength 1 shown along this y-axis and extinction at wavelength 2 shown along this x-axis, then we can represent, let's say, the extinction for protein A as a vector in this space. So it might have extinction along wavelength 1 of this, and at wavelength 2 this. All right. Now let's say that we have another component, component B, shown by the blue vector. That vector is exactly collinear with the red vector. And what that means is that there's absolutely no way that we can distinguish component A from component B. This matrix is by definition singular. And the mathematical way of describing that is that its determinant is 0. Well, so what we need is a non-singular matrix. In other words, we need those vectors to be not collinear. But the real question then becomes, how nonlinear do they need to be? And a colleague of mine named Shay Pedrick got very interested in this question when we started doing some sedimentation velocity multi-signal experiments together. And he came up with a metric that allows us to, that has some predictive value in figuring out whether a given matrix will be non-singular enough to basically allow us to do a reasonable multi-signal sedimentation velocity experiment. So let's look at this Cartesian space again, and let's say we have a vector for our protein A, and so its extinction properties look like this, and the extinction properties for protein B look like this. Well, if we take vector A and we add it to vector B, and vector B and add it to vector A, then we get something that looks like this. It's a parallelogram. And if we take the area of that parallelogram, that's actually the determinant of that matrix, basically. Okay. Now, the determinant by itself is actually not very descriptive. We need a way to normalize it. So what we're going to do is take the length of the red vector and the length of the blue vector, and we're going to multiply them, and that's kind of like getting this area here. Okay. So this area, and if we divide the determinant by the product of these vector lengths, we get this quantity called d norm. And d norm is the quantity that Shay dreamed up that allows us to figure out whether our determinant is essentially large enough for us to go forward with a sedimentation velocity multi-signal experiment. After a lot of simulation, we determined that we want, for typical proteins and signal-to-noise ratios, we would want a d norm of greater than 0.065%. Or sorry, 0.065. And so you'll find actually that if you're looking at protein-protein complexes, that this is fairly easily achieved. In fact, I have to look pretty hard for proteins, protein pairs that don't achieve this. Okay, so now that we've talked a lot about the theory of performing the sedimentation velocity experiment, let's go forward and talk about planning and performing these experiments. So questions for planning: is the norm large enough? Is the complex stable enough? Basically, we want the complex to be populated throughout the entire course of the experiment, which means we want K-off to be slow, or we need to add something in significant molar excess for us to be able to keep the complex populated during the course of the sedimentation velocity experiment. What signals will be used? Are the concentrations going to be within the limits of detection? So these are basically the limits of detection that we want to have for absorbance and interference optics respectively. And we want to be able to build all the knowledge of the system into the strategy. And I think I'll be touching on all these points through examples in the next few slides. One thing I do want to add here is that the signal increment for at least one signal must be known well. We must know extinction information before starting to perform the analytical ultracentrifugation experiment for at least one signal for all components. Okay, so that might be that you know the extinction coefficient at 280 for one of your components very well from having either determined it empirically or calculated it. Okay, so complex stability is important. We need it to be fully populated throughout the experiment. This is not a problem for systems that have a slow K-off, but for systems that have a fast K-off, which is probably most of them, because slow K-off means probably 10 to the minus fifth per second. And so there's not a lot of protein-protein complexes that would be interesting to study that have that slow a K-off. At any rate, with a fast K-off, basically what we need is we need to choose concentrations that fulfill our requirement. Usually this means having a significant molar excess of one of the components, and most often this is best achieved by having the smaller of the two components in molar excess. And that's just so that we keep within the signal limitations of the instrument. The choice of signals, basically we want to maximize the norm. If we're doing an absorbance-only experiment, we'll probably want to look for differing ratios of tryptophan to tyrosine, which will allow us to do a 280 to 250 experiment. In other words, monitoring the experiment at 280 nanometers or 250 nanometers. And again, I'm talking about proteins here. For interference absorbance, we look for the ratios of mass to tryptophan, because the signal increment in the interference optics is basically directly proportional to mass. So we need, basically, if we have different mass to tryptophan ratios, then we can monitor interference optics and absorbance to 280, and that should allow us to have differing, basically a large D norm. Also, we want to choose signals so that we're collecting at peaks or valleys. So this is a typical protein absorbance profile here. You would want to, if you were collecting an absorbance-only experiment, you would probably want to collect at the two wavelengths I've highlighted here, 280 and 250. The reason that you want to collect at peaks or valleys is that there's a little bit of slop in the Beckman optics with regard to wavelength accuracy. And so if we miss the mark a little bit, say we end up maybe right here, then we're not changing the amplitude of the absorbance units very much. But if we're on a shoulder and we miss the mark a little bit, then we're changing our amplitude by a lot. Okay. All right. For a two-component interaction, usually 3 sedimentation velocity experiments are necessary. Component A by itself, component B by itself, and the mixture of AB. The first 2 experiments are necessary because we want to establish the component S values, and we also need to refine extinction information. So remember, we know extinction information for one signal. we're going to determine extinction information for the other signal in the centrifuge. That's what these first two experiments are for. The third experiment then, of course, is for stoichiometry. All experiments need to be monitored at the same signals. The number of signals must be at least 2 for our two-component interaction. It can be more than 2, though. If possible, it's best that we use the same concentrations in experiments 1 and 2 that we used in 3. This is a very powerful control, and this will come out in the example analysis that I'm going to do. So my example here is a published example. This is human lactoferrin interacting with a bacterial protein called TP34. TP34, we did an ITC experiment, and we got this ITC result over here. And basically what I'm showing you is that this has 2 modes in it, and obviously there were 2 binding events. We found that the molar ratio of interaction was 2 to 1, so we wanted to know what the real stoichiometry was, and we also wanted to confirm this, because basically this is a very highly unanticipated result, and we wanted to be able to verify that using a multi-signal sedimentation velocity experiment. Now, I should tell you that human lactoferrin was from a commercial source, and it was expected to be glycosylated. Therefore, because the interference signal increment is due to the mass, we didn't know how much mass there would be for human lactoferrin because we didn't know the extent of glycosylation. We therefore used the method of PACE to figure out the extinction coefficients for TP34 and human lactoferrin at 280 nanometers. And so these are the values that we ended up getting. We chose to do interference and absorbance at 280 because we felt that the mass to tryptophan ratio was different enough. This was actually performed back before we had denormed to lean on, so we were just looking at mass to tryptophan ratio. And we basically did those 3 sedimentation velocity experiments that I outlined in a few slides ago simultaneously in a single rotor at 20 degrees at 50,000 RPM. Okay? And so here are the results. Here is the data for TP34. The top shows the absorbance data. The bottom shows the interference data. And again, these were from exactly the same cell. Data were collected concurrently during a single run of the centrifuge. The human lactoferrin alone is shown here. Again, absorbance on top, interference on the bottom. And then the mixture is shown here. And again, you can see that we have 2 boundaries, one there and one there. So we had a large molar excess of TP34. That's the slower boundary. And then the faster boundary is the complex between TP34 and human lactoferrin. So now that we've collected the data, we need to do the analysis. And so basically I'm going to have to skip over a lot of steps because I don't have time to really go through all the nitty-gritty details of understanding how to import data into SEDFAT. However, there is web documentation on how to do this. What I've done is loaded 2 experiments, and they're called Experiment 1 over here on the left and Experiment 2 over here on the right. This on the left is the absorbance experiment, and we're calling that wavelength 1. Interference is over here on the right, and we're calling it wavelength 2. The program calls it wavelength, even though, of course, what we're collecting here is interferometry. I want to point out that all of this is done in Peter Schuck's program, SEDFAT, and he's the one who came up with the methodology to do this computationally. Under the model in SEDFAT, we choose multi-wavelength discrete continuous distribution analysis. The program will ask us, is 2 wavelengths correct? It's detected 2 experiments, so it thinks that there are 2 signals. We say yes. And then it asks if everything is in the standard configuration. In other words, is set 1 wavelength 1? Is set 2 wavelength 2, etc.? We say yes. We also need to take particular care to have the right experimental parameters in there, and that's accessed from the main menu in SEDFAT. For the absorbance experiment, I've put in this information, and this basically I'm allowing the baseline and PI noise to refine. The same information is input for the interference experiment shown over here, except we're also allowing radially invariant noise to refine here, and that's just standard for interference data. What's most important actually probably here is that I'm not refining the menisci. An empty check button here in SEDFAT means I'm not refining the value of the meniscus. I set the meniscus when I loaded the data in. I'm not going to refine it yet. And that's because meniscus refinement in the early stages would cause us, basically cost us a lot of time. So we want to keep that parameter fixed, and then we'll refine it at a later time. Now, here is the global parameter window in SEDFAT, again accessed through the main menu. And it looks like a really, really horrible mess, but it really isn't. It's organized very well, and we're actually only going to deal with a subset of the windows here. So, for example, we're not going to use the top part because that's for discrete species. Here we have the CK of S information. Here's the extinction information. And here is some CK of S properties that we'll have to designate. Okay, so looking at the extinction part first, for this single species, again, this is component A, TP34 by itself that I've loaded here. So I only have one species, and that's termed chromophore in the parlance of SEDFAT. So I only have one chromophore, so my matrix only has one row in it, really. It actually has two rows, but those are going to be zeros in the second row. Now I've input the information that I know, which is the extinction at wavelength 1, which is the interference, or I'm sorry, which is the absorbance. I don't know the signal increment for interference for TP34, but a good guess is 2.75 times the molar mass of the protein. Note that I have clicked on the refinement of this value. One of the big points of doing this analysis by itself is that we need to refine this number and figure out what the extinction is at wavelength 2. Okay? So my next task is to figure out what properties the CK of S distribution is going to have. So I've turned on this segment by checking this checkbox here. Now, those of you who are familiar with SEDFIT, this top row will look pretty familiar to you. I'm giving it a resolution of 50. That means that I'm going to be considering 50 sedimentation coefficient values, and their bounds are 0.2s and 10s. I've input a frictional ratio of 1.5, but again, I'm not allowing that to fit for the same reason that I'm not allowing the menisci to fit. Now, down here, we have these two spectra. The spectra are actually the CK of S distributions. So I told you that in the end, two CK of S distributions are going to have to overlap in one segment. And so, because we have two signals, it will allow you up to two of those CK of S distributions. However, we only have one component here, TP34, or protein A. And so, I've only turned on one spectra, and now we have this kind of what I would call a stoichiometry matrix over here. What I'm telling it is that by putting in a 1 here and a 0 here, I'm telling SEDFAT that spectrum 1 is going to represent 1 to 0 complexes of chromophore 1 to chromophore 2. Now, of course, there is no chromophore 2 here, but that is still important to put in, 1 and 0. Okay. Next, we want regularization, so we click on this, and we want the distribution to be normalized, so we click on that. All right. So then we would click OK in that window, and we would allow a global fitting of the basically of the distribution. And now we get a single CK of S distribution down here that represents TP34. Now, we can immediately see that this is pretty much a single species, but the only thing that we're really refining in this case was this value right here, which is the signal increment for the interference optics. Again, that's wavelength 2 shown there. And so that's our initially refined value. It was a little bit higher than what I thought it was, but it seems reasonable. Now again, throughout this we're going to have to assess whether the fits are good or not. And I'm going to delay this discussion on that until a later point in this talk when we'll sort of globally go over whether the fits are good or not. But take it from me that this fit at this point is pretty good. Now what I need to do is go back in the experiment parameter windows and turn on the menisci. Now I want these to refine. Now that we have found a good value for the signal increment for the interference optics, we can let other nonlinear parameters refine. We would also want to do that for the frictional ratio, and I forgot to put in the slide for that. All right, so now we do another fitting, and this is the distribution that we get. So what did we learn from this distribution? We learned that if I integrate over the entire distribution, I find that the concentration of inputted TP34 is 15.6 micromolar. I'm going to want to note that and write it down somewhere. I also found out what the signal increment, the final refined signal increment for TP34 at wavelength 2, that is for the interference optics, is, and it's 60,887.3. So those are the two extremely valuable pieces of information that I got out of this experiment, and I need to do that also for human lactoferrin. Okay, so this is the data loaded into SEDFAT for human lactoferrin. I'm not going to go through the process again very thoroughly because it's very much analogous to what I did for TP34. We would want to have our known extinction information for wavelength 1, the absorbance, and a guess for wavelength 2. We're refining that. Okay. In this case, I think that there's going to be a little bit of aggregate present, so I put 15 as the maximum sedimentation coefficient. But again, since I'm defining this human lactoferrin as chromophore 1, I'm only doing 1 to 0 complexes under the spectra here. This is, after all the fitting, the distribution I get for human lactoferrin. And so as you might expect for basically a commercially available protein that you would buy from Sigma or something, it's not perfectly pure, but most of the material is sedimenting here at the 5.5S position. So what did I learn here? I learned that the total amount of input signal or concentration of human lactoferrin, if we count all of this as human lactoferrin, is 1.8 micromolar. And I also learned the refined value of the signal increment for the interference optics, which is 232,606. All right. So now, finally, the experiment that we need to analyze is the mixture of the two. So again, you can see that there are two boundaries present, and I've loaded wavelength 1 as the absorbance data, wavelength 2 is the interference data. Now, I'm going to input the extinction information. This extinction information I know. Now here I'm defining chromophore 1 as TP34, but now I have two chromophores present, two species present, so chromophore 2 is going to be defined as human lactoferrin. I've inputted the known extinction coefficient of both human lactoferrin, TP34 and human lactoferrin here, and then the refined values for the signal increment and the interference optics here and here, respectively, for the two proteins. Note that none of these checkboxes are checked because I don't want this information to be refined. These need to be fixed. Okay, so again, chromophore 1 is TP34, chromophore 2, human lactoferrin. Okay, now don't be too put off by this. I'm actually splitting S space into 3 segments here, from 0.2 to 4.8S, from 5 to 7S, and from 7.2 to 15S. This is just a matter of convenience for me to be able to look at different regions of S space with different parameters if necessary. But basically, this is very similar to what I did before with only one segment. Now I need to define 2 spectra. Spectrum 1 is going to be examining 1 to 0 complexes of chromophore 1 to chromophore 2. In other words, this CK of S distribution defined by spectrum 1 will be looking only for TP34. So that CK of S curve will only represent TP34. The second CK of S curve that occupies the same S space will look for 0 to 1 complexes of chromophore 1 to chromophore 2. In other words, it will only report on the amount of human lactoferrin present. This matrix is the same in all 3 segments. Okay. All right. So after we do the global fitting, this is the result that we get. You can see now that we have this region here of co-sedimenting human lactoferrin and TP34, and the information on stoichiometry that we wanted is in there. However, I'm not going to tell you what the answer is just yet. I need you to take a breath and delay the gratification for just a moment, because what we're going to do is go over goodness of fit criteria for these experiments. And so, and again, these would apply to all 3 experiments that I've just shown you. So basically, the first thing that we want to assess is chi-squared and RMSD okay? The RMSDs are shown at the top of the windows here and here. For my absorbance and interference optics, these are actually very good numbers. The global chi-squared, global reduced chi-squared, is shown here, and that is 0.26 in this case. It's difficult to give you a recommendation for an absolute value there, but you know, if this basically, if the assumptions of noise that went into the experiment are correct, then this should be, you know, somewhere between 5 and 0.1 or something. But anyway, this value of 0.26 has me not worried at all. Are the residuals random and near to 0? And for the most part, we can say that that's true. There may be a little bit of systematicity in the absorbance residuals, but for global analysis, this looks really good to me. Do the total concentrations of components match what we put in? This is where that concentration information that we got from experiments 1 and 2 comes in handy. If I integrate under the entire distribution for this 2-signal analysis here, then what I end up getting is 15 and 2. Now, I thought that the input was going to be 15.6 and 1.8. So those errors are pretty small. I'm thinking that this was actually quite a good recapitulation of the input concentrations. And so it looks to me like the analysis is working quite well. All right, so now we get to gratify ourselves with understanding the answer. If I integrate just under this region, I get a concentration of human lactoferrin of 1.3, and of TP34, I get 2.7 micromolar. So that is scarily close to 2 to 1, or 1 to 2, however you want to think about it. 1 human lactoferrin for every 2 TP34s. And when we look at the hydrodynamic information that we get from knowing that the sedimentation coefficient is 6.5 roughly here, we can understand that this is truly 1 human lactoferrin for 2 TP34s. The molar mass that we get out of that is consistent with that true molecular stoichiometry. Okay, so I just wanted to show you that this is an example from a paper by Hauptmann et al. This was performed in the lab of Peter Schuck. And you can do this with 3 signals, and so this was done with 3 proteins, a protein called LAT, one called SOS, and another called GERB. The LAT was labeled with rhodamine, and then I believe absorbance of 280 and interference were also monitored. And so 3 components, 3 signals, and you can see that you can actually get fairly good resolution and have some guesses about stoichiometry here. Okay, so now in the last few minutes that I have, what I wanted to do was to talk to you about a system where the multi-signal sedimentation velocity failed horribly. And this is a case, again, I think that these cases are fairly rare, but in this case, the D norm was very, very small, 0.016. So I'd actually hunt very hard to find something that bad. But this actually really compromises our ability to spectrally decompose the C of S distribution into CK of S distributions. So if I do what I would call an unconstrained analysis, which is exactly what I just showed you for TP34 and human lactoferrin, I get an analysis like this for TP34 interacting with a different protein called bovine lactoferrin. From ITC experiments, we understood that the stoichiometry of this interaction was different. It was probably 1-to-1. At least the molar ratio of interaction was 1-to-1. And so we wanted to, again, confirm this using the multi-signal methodology. Well, when I do the unconstrained analysis, this is what I get. There are some big problems here. Okay. Actually, let me go back. First of all, I want to point out that bovine lactoferrin has a sedimentation coefficient by itself of about 5.2s. Well, SENFAT is telling me that all the material below 4s is actually bovine lactoferrin. That's physically impossible. So that's a sign that something has gone horribly awry here. SENFAT is also telling me that the molar ratio of TP34 and bovine lactoferrin is, I don't know what this is, it's probably something like 5-to-1, but that doesn't make hydrodynamic sense for proteins of this size. And so these 2 criteria tell me that something is really, really wrong. The other thing that tells me that something's wrong is that the known input concentrations shown here do not match the calculated output concentrations shown here. And these are actually rather large differences, and so in some cases up to something like 20 to 30%. So we basically, this is what failure looks like. This is a complete failure from the standpoint of multi-signal sedimentation velocity, and that's because our D norm is just too small. All right, so is there any hope for rescuing this kind of experiment? Actually, I wouldn't ask the question if the answer wasn't yes, so yes. We can introduce a new constraint. In other words, that table that I showed you, we know the input concentrations. We can constrain the analysis to have, or at least to try to cleave to those concentrations as closely as possible. This is called mass conservation multi-signal sedimentation velocity, or MSSV. Note that when we use this as a constraint, we're losing it as a quality assessment metric. Okay. And there's a paper about this in PLOS One if you want to check that out. And again, I refer you to that paper for a lot of the details about this. Again, we're getting a little short on time. But what you would do to do this analysis, in the global parameter window, there's a little section that has a with total concentration prior knowledge. So we click on that check mark. Then we say, SEDFAT, I want you to enforce the input concentrations, which I inputted by hand here, to within whatever percentage, and I input 5% here. I figure my pipetting technique is good enough to know the concentration plus or minus 5%. So what I've done then is basically told the analysis, yes, I need to be constrained to plus or minus 5% of these known input concentrations. Now, when I do the analysis, it turns out that there's still this problem of there being not enough spectral discrimination to say that the stuff that is at 2s is really TP34 and not bovine lactoferrin. Again, the molar ratio here turns out to be wrong as well. So we need to add in another constraint. And again, this is another example of adding in information when you know it, and you know it for sure. So the way to do this then is to split S space up into two spaces. And I showed you how to do 3 spaces. This is 2 spaces here. So I have one segment going from 0.2 to 4s here. Now what I've done is I've told SEDFAT that only TP34 gets to be in this space. Bovine lactoferrin is too big to be here. We shouldn't detect any there. So I've turned off the second spectrum. And I've said, SEDFAT, I want to see only one CKS distribution here. And it should be 1 to 0 complexes of chromophore 1 to chromophore 2. In other words, only TP34. But I've allowed segment 2 to have both, shown here. Okay? So by building in this information of knowing that TP34, only TP34 will be detected between 0.2 and 4s, I'm helping the analysis out. So I'm constraining mass, and I'm constraining this smaller segment sedimentation coefficient range to contain only TP34. So now, and I have to apologize, I actually wrote in incorrectly human lactoferrin there, so let's scratch that. That's going to be bovine lactoferrin. A little revision here on the fly. So now you can see that due to my constraint, we have only TP34 down here at small s values, and now we have a co-sedimenting complex of bovine lactoferrin and TP34 at about 5.8s. The molar ratio turns out to be 0.76 to 1. It turns out that I probably didn't have enough TP34 in there to keep that complex totally occupied during the course of the sedimentation velocity experiment, and so this caused a slightly strange molar ratio here. However, what's interesting to note is I actually went back and did this experiment again. It turned out, this is back before I really appreciated the need for denorm, but I found that if I collected the data at 2 signals, 280 and 250 nanometers instead of interference and 280, which is what this experiment is, then I actually get a much better denorm, something in the range of 0.2 or something like that, or not 0.2, 0.02. Much better spectral discrimination. I can do the unconstrained analysis, and I end up getting, under identical conditions, the identical answer. So, and I mean scarily identical. Okay, so the idea here is that the mass constraints and the sedimentation coefficient constraints that I put on this analysis actually worked beautifully. Okay, so to conclude, basically I find multi-signal sedimentation velocity to be a very powerful means to discover the stoichiometry of macromolecular complexes. It uses the capabilities that are found in current Beckman instrumentation, I think I've impressed on you that careful experimental planning is absolutely required. It can be done in a single run at the AUC. I think all of the experiments I showed you were done in a single run of the AUC, which basically means 24 hours of running, maybe less. And even in these cases where we have challenging spectral discrimination problems, we can get excellent results out. So in the end, I do want to acknowledge the fact that I'm not the inventor of this technique by any stretch. I'm just the conveyor of it. And so Peter Schuck has invented this CK of S methodology way of looking at multi-signal sedimentation velocity data. And if you're interested in doing this, if you haven't already done so, I encourage you to download SEDFAT, which can be found at the link shown here. If you liked how the graphs looked on here, virtually every two-dimensional graph shown here, including the ITC data, was made with my program, Gussie, which you can download at the link shown here. Just a few acknowledgments. Peter, of course, the proteins that I worked with, bovine lactoferrin, human lactoferrin, TP34, were all provided to me by Michael Norgaard's lab here at UT Southwestern, and in particular a brilliant protein chemist named Ranjit Dekka. And also I want to reiterate that Shea Pedrick was a major motivating force for getting the D-norm metric going. So with that, I'm going to turn the floor back over to Chad Schwartz, and I'd be happy to take any questions that you might have.
Great. Thank you for that terrific presentation, Dr. Brautigam. Before we get started on the question and answer session, I would like to remind our audience that you can submit questions by typing them in the Q&A box at the lower left of the presentation window. Several good questions already came in. Our first question is, can samples and complexes that do not absorb in the UV or visible spectrum be analyzed?
My initial thought about that is probably no. And the reason is that you're going to need at least two signals to be able to do this type of experiment. If you don't absorb at all anywhere in the UV or visible region, then you've got a case where you've only got one signal available to you, and that is the interference optical system. But because you only have the one signal, I do not think that this experimental strategy would work for you.
Great. Thank you for that answer. Our next question is, can this technique be used preparatively, and what is the dynamic range of AUC?
I think it would be difficult to use this technique preparatively, and that is because, first of all, the early parts of the analysis that I'm talking about, the so-called experiments 1 and 2, are very dependent on having pure components. And so if you had a mixture of components that you were trying to isolate or something, that would not be a good strategy. Analytical ultracentrifugation in general is not considered to be a preparative methodology because basically retaining the samples or getting the samples after the experiment is problematic. There would be mixing as they would be taken out of the rotor. Even if there wasn't, basically at the end point of the experiment, everything is pelleted at the bottom of the centrifugation cell, so there would actually be no separation or preparative value there. So I don't know of anyone having used this as a preparative technique. Now I will say that I do know of one case where someone has done, I believe, a sedimentation equilibrium experiment, and then taken the supernatant from that experiment, which is done at a much lower gravitational field than a sedimentation velocity experiment, and then taken that material and done a dynamic light scattering experiment on that. Now that actually has a huge advantage because by sedimenting for several days in that experiment, you have really ridded yourself of contaminating, let's say, dust or something like that. And so you could, the potential for getting a very clean dynamic light scattering signal out of the supernatant from that kind of experiment is there, and you might consider that to be preparative.
Okay, great. Thank you for that.
I missed the dynamic range part. Sorry. The dynamic range was presented on one of my slides. You need, because you're going to be using the absorbance optics for at least one signal, you need to be within the absorbance units limits for that. So generally I wouldn't want to be below, say, about 0.1 absorbance units, and I wouldn't want to be much above 1.0 absorbance units. One does have to keep in mind that in the Beckman centrifuge, usually the centerpieces have a path length of 1.2 centimeters. So you're actually going to have 1.2 times the value you would have in a 1 centimeter path length. Beckman also sells centerpieces that are thinner, and that actually allows you to explore a wider dynamic range. So they are about a quarter of the thickness, so you could go up to an absorbance of roughly 4 in that case if you needed to.
Okay, thank you for that. Our next question is a method question. If you collected absorbance 280, absorbance 250, and interference for a 2-protein complex, is the use of all 3 of those optics best, or is it better to focus primarily on the absorbance 280 and the interference? And then the question follows up by asking, does using more signals make the analysis longer?
Okay, so taking the first part of that question, let's say you did 3 signals for a 2-component problem. I've actually done that a couple times, and if you look at the references that are cited in this talk, you'll see that I have published data using that strategy. The question really becomes, where is the information content of the experiment? And so the 2 signals that give you the highest D norm are basically going to be the ones that are contributing most to the analysis, to your ability to discriminate between the 2 components spectrally. Okay, so if you just did the analysis with the 2 signals that gave you the highest D norm, that would be perfectly adequate. Now, if you collected all 3 signals, it doesn't really hurt to put in all 3 from the standpoint of spectral discrimination, and it will probably help you with regard to basically the S resolution that you get out, because given that you have collected 3 signals, if you leave out one of the signals, then you're leaving out actually part of the evolution of the concentration profiles. So I would encourage you, if you collect 3 signals, go ahead and include them. Does it slow down the analysis? Yes. It has the potential to, because you're including more data, and more data means more processing time. Now, is that going to be really significant? I don't think so. It would, processing time would not scare me away from doing 3 signals, and in fact, if you were really judicious about the range of values that you use, there would probably be not a huge difference in the number of data points from a 2-signal analysis anyway. So if you do go through the pain to collect the 3 signals, I would use all 3 signals and really not hesitate to do so, just realizing that there's a strong possibility that most of the spectral discrimination is actually probably only coming from 2. Okay, I think I've addressed both parts of that question.
Great, terrific. Unfortunately, we're out of time today. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, Beckman Coulter, and of course, Dr. Chad Brautigam, for making today's educational webcast possible, which will also be available for on-demand viewing through beckmancoulter.com. Thanks again. Goodbye.